Welcome once more. We are now going to look at the life cycle of a flowering plant. I'm sorry, there's no way of running away from these life cycles. Now, you will realize that we have covered a lot about, or rather you know a lot about plants. Let me start from a region that you are aware of. I will start with the plant that you normally see. This is the plant that you see standing outside, and that plant is the sporophyte because it is a product of an embryo which was a diploid structure. Please note here, sporophyte is written here. That means all of this here is diploid and gametophyte is right here. We are very familiar with most of the sporophyte. Now when we go to the flower, we all know that the flower, most flowers, complete flowers or perfect flowers, have male and female. That is the male part and this is the female part. I will not bother you with that. Now this is the male part. We have looked at the pollen sac when we're looking at the pine, and we looked at how it matures. The maturity of this pollen is not very complicated. It is mainly differentiation in the anther to give us diploid cells, and these diploid cells will undergo reduction division to give us myospores or haploid cells, which we call pollen grains. We'll come back to the development of the pollen grain later. So you are familiar with the pollen grain. Let me come to the female part. You are familiar with these parts, the stigma, the style, and the ovary. What is important here is the ovary. The stigma is just to receive, the style is just to direct to the ovary. So mainly when we are talking about the life cycle of a flowering plant, the detail will be in what happens in the haploid state, in the gametophyte. This is the state which we do not readily see. So there's a lot of stuff you will not know because we are not even going to be looking at it under the microscope. We don't have that leeway. Let us go to something you have met before. We have the microspore. Please note, after reduction division does not happen again. It only happens once. Here, when the pollen grain is formed, and here, when the embryo sac is produced. Maturation of the pollen grain is simple, it's very similar to what you saw in pine. We end up with the same products, the tube nucleus and the male gametes. They are not flagellated, hence the pollen tube that will carry it to the embryo sac. What is most demanding for us here now is the maturity of the embryo sac. By maturity, I mean what happens to the haploid cell to be able to give us the egg that will give us the zygote. The following diagrams might be a bit confusing, or they might look a bit detailed, but you can draw them by hand. Let me skip this for a while. I'll come back to this. I want us to look at the maturation of the embryo sac. We have a diploid cell before we enter the gametophyte. This diploid cell undergoes meiosis. These are the stages of meiosis that you learned in year 1, and you end up with 4 cells that are haploid. I've shown you metaphase where you have the dyad, metaphase 2 until you get the the linear tetrad. We call it linear because they're in a row. Tetrad because they are 4. So here we have the 4 myospores. You remember in the pine, there was some... Ooh. Sorry sis, I'll call you back. You remember in the pine that there were some cells disintegrated. But before we go to the disintegration, let us look at the orientation of this line. This side is the micropylar end, where we have that opening. This side is what we call the distal end. Now, note that the 3 cells that are going to disintegrate are the 3 cells close to the micropylar end. This is similar to what we observed in pine. Now, once this cell remains alone, it becomes bigger. So we have disintegration of these 2, and this one enlarges. Let's go on and see. Okay, this one is enlarging, and after it has enlarged, it then continues to divide. But this division is mitotic, no more meiosis. So mitosis, each time it happens, we get 2 cells. So here we get 2 cells. The 2 cells, this one will divide, that one will divide. We end up with 2 cells this side, 2 cells this side. That is not the end in the maturation process. These 4 cells continue to divide such that above the equator we will have 4 cells. Below the equator, which I drew there, we have 4 cells. Now, these cells that are at the distal end are 3 of them come together, and these are the antipodal cells. They come together and they die or they disintegrate. 1 will remain, and that one that remains, it migrates to the equator. On this side we have these 4 cells. The 4 cells, all of them develop. There is no dying on the micropylar end. 3 remain on the micropylar end. 1 moves to the equator and it joins with the one that came from the distal end, and when they join, they become diploid. This is not the sporophyte. This is the nucleus that will give us the endosperm. On the side of the opening, one cell becomes bigger than the others. This is the egg, and the other 2 are just there to keep it company. We call them the synergies. Please note that they're on the side of the opening. When fertilization takes place, when fertilization takes place, only this egg cell will be important and this polar cell will be important. These 2 are just here to guard this egg until that time, and afterwards they don't play any part. So once we get these cells together, we say the embryo sac is mature. What does it mean to say it is mature? Mature means it is ready to be fertilized. So these 7 cells are all, most of them are haploid, except for those that I've shown you have combined. These 7 cells are the female gametophyte. Note that the gametophyte is no longer a plant we can point at. It is just a number of cells. We have the egg, we have the 2 synergids, they are haploid. We have the polar nucleus, which is diploid. We have the 3 antipodals, which are going to die anyway. But before they die, there will be 7 cells. But the important cells are mainly these cells that remain here. Now, this maturation to give us 7 cells is not always the case. Sometimes the maturation process is slightly different. This is the diploid cell. It undergoes reduction division, and we end up with these 4 cells of the linear tetrad. We are used to these 3 cells at the antipode. Well, they're not like this at the beginning, because that time I put a line through here. But this time I'm not putting a line through here. I'm just dividing them such that there is 1 here, then I put my equator. These 3 come together, and this one remains. Now when these 3 come together, they will fuse. Remember the 3 that we had earlier disintegrated. These fuse and they remain. There are 3N. They have a 3N nucleus, and this remains with the haploid nucleus. This then goes through the mitosis I told you about, such that we continue 2 cells, we have 4 cells. But note these 4 cells, 2 and 2. This one has a big nucleus, a 3N nucleus, and the other one has a haploid nucleus. Okay, let's continue. The division continues, like I told you earlier. These 2 will divide such that above here we will have 4 cells, and over here we'll also have 4 cells which are haploid, and here we have 4 cells which are triploid. Now what happens here? One nucleus, as I told you earlier, migrates to the equator. 1 nucleus from here migrates to the equator. This results in this nucleus being 4N. Whereas earlier we had a nucleus that was 2N, now we have a nucleus that is 4N. The antipodals remain there 3, and here we have the egg apparatus, which are the synergies and the egg. Once we have the antipodals that are 3N, they will disintegrate. It really doesn't matter. But the important thing is the 4N nucleus is the endosperm nucleus, and this apparatus remains the same as we had before. Now let us see what happens. This is the end of maturity. Now let us see what happens in the life cycle. Yeah. We're going back to a diagram I skipped, and this is the diagram I skipped. In this diagram, what I have here is what I've already explained, what happened in the plant. We have the stamens, the carpels, etc., etc. We have a megaspore, we have a microspore. Okay, here there is no problem in the male. We have the pollen grain. The problem or the confusion might happen here. The female that developed is what we call the embryo sac. The embryo sac has the endosperm, the endosperm nucleus, which is either the 2N, which we saw earlier, or the 4N, which we saw later. Now, the male will give us the 2 sperm cells that we saw. Normally, we only care about fertilization of the egg. But now we are concerned also about what happens to the nucleus of the endosperm, which we saw the 2N and the 4N. The sperm will fertilize the egg. Fertilization of the egg by the sperm is the fertilization we are used to. It gives us a 2N zygote. Fertilization of the sperm with the endosperm, with the endosperm nucleus, will give us an endosperm, which the zygote needs for food. If it is a 2N, this endosperm will be 3N. If it is a 4N, this endosperm will be 5N. Most plants have the 3N endosperm, but there are plants that have the 5N endosperm. In the diagram I showed you, this organism here is Carex. It could be anything you like, but here, this is Lilium. Lilium is a lily, and Lilium has a 5N endosperm. So if somebody asks you about the life cycle of a flowering plant, they have to specify to you this is the life cycle in diagram form. But for you to complete this, they have to tell you whether this plant has a 3N endosperm or if it has a 5N endosperm. That means they are asking you about, mainly they're asking you about how this embryo sac matures. Please practice the diagrams I gave you earlier in a way that will be easy for you to reproduce in a test situation. And always show which side is the micropylar end, which side is the distal end. This is the life cycle of an angiosperm in words, and this is the life cycle of an angiosperm with diagrams. The best thing to do is, you can use words for things that are not complicated, but when it comes to stuff like this, please use the diagrams when it comes to the embryo sac maturation. Over here, this is the half flower, and you are being shown a developing pollen and a mature embryo sac. This kind of tells you the duties of all the things that are there, that this has to go through the micropylar end to deposit the sperms in the mature embryo sac. So this is the mature embryo sac with the antipodals 3 over here, the endosperm nucleus, the egg apparatus over here. Let me take this opportunity to make you look at this. This is the pedicel where all the things are attached. You can see that these are the sepals that are attached here. They'll form an outside circle. These are the petals that are attached here. They form the next circle. Then these are the stamens. They are attached here. They give you the next whorl. And in the center, we have the gynoecium. This elongated axis I was telling you happens in that the pedicel will not be flat like this if the axis is elongated. If the axis is elongated, the pedicel will be raised and domed like that, such that these things don't enter at the same level. They will enter at various heights from the base. That is an elongated axis. Thank you. I hope this clears all problems you might be having with classification, flowering plants, and welcome to angiosperms. This part of the course is the easiest because this is what they teach us at school, because we can all see plants outside. It is just the terminology that might be different. Okay, thank you.